As a scientist, I think it's curiosity that's really the main driving force. You get to be curious and to ask questions, and that's what I find very intriguing. I'm Melissa Grevert. I'm a beamline scientist and I work for the EMBL here on the Hamburg site. So with our technology, we help users, but also EMBL scientists, to take pictures or visualize the very small building blocks of life. So in a cell, we have stuff like proteins, DNA, fats, and sugars. And these are the items that actually make our cells work. Structural biology is quite a bit about the proteins, so they're really the ones that do the action. They're the enzymes, they chop up stuff when you digest something, but also antibodies are proteins, so they're the ones that help our immune system attack the invaders when we get sick. So that's the ones that most our users are interested in taking a closer look at. You can tell from the surrounding it's not your typical lab where we're at. So we're at a synchrotron, so that's a particle accelerator where we have electrons going around in a circle really fast, and these produce X-rays. So what we do is X-ray technology. So from here, from the bridge, we can actually see how the X-rays enter our hutch all the way in the back, so 80 meters. That's how far our exit tube is. So we generate the X-rays, and then they come inside the tube, and then they finally enter the hutch, where they'll be hitting our samples, and that's where we can collect the information that we need. So if you want to take a look at these small building blocks that make up life, there's different ways how you can do it. Our method is an advantage that we're measuring in solution, so we can watch the proteins in action. They're not being forced in a frozen state, or they're not being forced into crystal packaging like in other methods. So we can add stuff, we can change the temperature, we can change the pH, we can test drugs, and we can just see what happens when we're changing the conditions and watch the protein change and move its shape. So as a beamline scientist, I help develop the technology and bring it up to standard, but I also help the users that come and use the technologies. So now we're actually where the experiment happens. So this is where we're going to load the sample. So we put it in our robot. And then when we start the experiment, the needle's going to suck the sample and park it inside here. And then this is where the X-ray is going to hit the sample, and we can collect our image that we're looking for. So now once the sample's inside the robot and it's ready to start the experiment, which is pretty simple, the user just types in the information they want to have as metadata and pushes a submit button, and the measurement goes. So I guess one example I can use is what we did during the corona research, and we're trying to understand if we can find blockers that can stop the spike protein. The virus needs that protein to bind to the host cell, and that's how it incorporates or infects the host cells. So as we know, there are the antibodies that can be used as a medication if you have COVID, but they're really expensive to produce. So this was a way to mimic what the antibodies are doing in a cheaper way. So this was a good lead, and it's being further pursued. Of course, you get rewarded by seeing the science that your users are doing and seeing how the technology can help a whole wide range of people. So the impact that you have on the community is pretty nice, and that's kind of satisfying.